Hello friends, I am Suvarna Venkatrao Gaikwad, working as Assistant Professor in Department of Botany, Dada Patil Mahavidyalaya Karzan, District Ahmednagar. We are dealing with the T.Y.B.Sc paper 3rd, that is Genetics and Evolution. In previous lecture, we have seen introduction of genetics. In that, we have studied branches of genetics, applications of genetics, and genetic terminologies. Okay. In today's session, we are going to discuss about selection of experimental material, monohybrid cross, dihybrid cross, law of dominance, law of segregation, and law of independent assortment. Let us see one by one. First is the selection of experimental material. So Mendel selected garden pea plant as his experimental material. The scientific name of garden pea plant is Pisum sativum. So, the question arises why he selected only pea plant. So, behind this, there are number of reasons, such as the first reason is pea plants are easy to grow. Then, they require small space for cultivation. The next reason is duration of pea crop is of single season. So its many generations can be studied within short duration. The next reason is seeds are larger and easily germinate. Next reason is pea plant produces many seeds resulting in large number of offsprings. This facilitates maximum chances of expression of character. One of the most important reasons is that pea plant is a bisexual flower, which means both sex organs are present on the same plant. The next reason is pea plant shows self-pollination. So what is this self-pollination? Self-pollination means transfer of pollen from anther of one flower to stigma of another flower. Okay, on same plant. So this self-pollination is one of the important reasons for selecting pea plant as an experimental material. Then, due to large size of flower, cross-pollination of pea plant is also possible. Cross-pollination can be done by first removing anthers from the plant, then collecting pollen from a different plant, and finally transferring pollen to stigma of individual whose anthers have been removed. So in this way, artificial cross-pollination can be done. Most important feature of a plant is it shows 7 pairs of contrasting characters. So these are 7 pairs of contrasting characters. In that there are 7 characters such as seed shape, seed color, flower color, pod shape, pod color, flower position and stem height. If you observe it, for each character shows two forms. Okay. For example, seed shape present in two forms. That is, one is round and second is wrinkled. So round form shows dominant character and wrinkled form shows recessive character. Now we have already discussed in previous lecture that dominant character is the character which appears in F1 generation and recessive character is the character which does not appear in F1 generation. So next, for seed color, yellow color is dominant and green color is recessive. For flower color, purple flowers are dominant and white flowers are recessive. Pod shape, inflated pods are dominant and constricted pods are recessive. For pod color, yellow pods are dominant and green pods are recessive. Next is the flower position. Axial flowers are dominant and terminal flowers are recessive. And finally, stem height. Tall plant is dominant and dwarf plant is recessive. So these are some of the reasons so that Mendel selected pea plant as experimental material. Okay, now we will move further. So the main important cross is monohybrid cross. If you look at the term indicates mono means single, that is cross between two homozygous individuals differing in single pair of contrasting character. Okay, just have a look at an example. This is an example of a monohybrid cross in that Mendel crossed pure tall plant with a pure dwarf plant. In this example, Mendel considered only single pair of contrasting character, that is single character, that is stem height. Stem height present in two forms, tall and dwarf. So, whenever we will study a particular cross of genetics, we have to keep in mind that there are some terms such as parents, gametes. So, this example indicates the steps. So, the pure tall and pure dwarf plants are the parents. Then the capital T and small t, these are the genotypes of the parents. Next, from capital T, we got single capital T as gamete. And further, when we cross capital T with small t, we get capital T small t, that is heterozygous tall in F1 generation. Okay. When these F1 generation undergo selfing, so selfing means what? Cross of F1 individuals with themselves. So when heterozygous tall plants were crossed with the heterozygous tall in F2 generation, we get three types of offsprings. What are these offsprings? Such as homozygous tall, heterozygous tall and homozygous dwarf individuals. So in this way, if we look at the phenotypes, there are three different types of phenotypes such as homozygous tall, that is the capital T, heterozygous tall, that is capital T small t, and homozygous dwarf, that is small t and small t. So in this way, if we get a ratio on the basis of phenotypes, so in this example, tall plants are 3 and dwarf plant is 1. So 3 tall and 1 dwarf. In this way, 3:1 is the phenotypic ratio of the monohybrid ratio. Next is the genotypic ratio. Genotypic ratio is based on the genotypic constitution. If we look at this example, capital T comes single time, capital T small t comes two times, and small t comes one time. In this way, genotypic ratio is 1 is to 2 is to 1. So in this way, Mendel considered this monohybrid cross by considering single pair of contrasting character. Okay, so we will move further. Next is the dihybrid cross. Di the term indicates di means two. So the cross between two homozygous individuals differing in two pairs of contrasting characters is called as dihybrid cross. If we look at this example, in parental generation, there are there are two individuals such as pure yellow round crossed with the pure green wrinkled. With that, in F1 generation, we get heterozygous yellow round plants. When these F1 generation individuals undergo selfing in F2 generation, we got 16 number of offspring. Out of that, 9 offsprings were yellow round, 3 offsprings were green round, 3 offsprings were yellow wrinkled, and 1 offspring is green wrinkled. So, when Mendel considered a cross with considering two pairs of contrasting character on the basis of phenotypes, and phenotypic ratio is 9 is to 3 is to 3 is to 1. So, in this way, a dihybrid cross can be studied. Next, we will move towards the principles of inheritance. So, Mendel put forth certain laws of heredity, and these laws are called as Mendel's laws of heredity or principles of inheritance. So these principles of inheritance are basically three. One is the law of dominance, second is the law of segregation, and third is the law of independent assortment. Okay, let us see one by one. First is the law of dominance. Law of dominance states that when two homozygous individuals are crossed with a single pair of contrasting character, then only one character appears in F1 generation, and that character is called as dominant character, and the other character which does not appear in F1 generation is called as recessive character. Let us take an example. In this example, Mendel crossed purple flowers with the white flowers. After crossing in F1 generation, all plants had purple flowers. Okay, so out of two parents in F1 generation, only purple flowers are there. So here, purple flower is the character appearing in F1 generation. So purple flowers are dominant, while the other character, white flowers, does not appear in F1 generation. So it is a recessive character. So this is a simple law. Then second law is law of segregation. Law of segregation states that when two alleles for contrasting characters are brought together in a hybrid condition, then these alleles remain together without mixing with each other, but they segregate from each other at the time of gamete formation. Let us study by example. In this example, there are two alleles such as capital T and small t. So these two alleles, they remain together without mixing with each other. Okay? These two alleles, they remain together without mixing with each other. But when these two alleles, they undergo gamete formation further, they segregate from each other and entered into separate gametes. Okay, so capital T enter into separate gamete and small t enter into separate gametes. That is, capital T and small t get separated from each other at the time of gamete formation. Now, as each gamete receive only one of the two allele, so this law is also called as law of purity of gametes, because gametes are always pure. So, this is all about law of segregation. Next, we will study law of independent assortment. So, Law of independent assortment states that when two homozygous individuals are crossed with two or more pairs of contrasting characters, inheritance of one character is independent on another character. Very simple law. Just take an example. The law states that when we cross two individuals considering two or more pairs of contrasting characters, at that time inheritance of one character is independent on inheritance of another character. For example, in this example we have taken the two characters, that is seed color and seed shape. So for seed shape, there is a round, capital R for round, small r for wrinkled seed shape, capital Y for yellow and small y for green seed color. Okay. So in F1 generation, these seed color and seed shape, they both four genes, they remain together. Okay. But when these F1 generation they form gametes, then capital R gets separate from R, small r, and they form new combination with the Y, such as capital R, capital Y. It forms yellow round. Similarly, capital R again forms new combination with the small y, that is green round. Similarly, small y get form new combination with the capital Y, that is yellow round, and again small r with the small y with, such as green wrinkled. That is seed color character and seed shape character. These two characters, these are not depends on each other, so that these two alleles they segregate from each other, separate from each other, and forms new combinations. Because of that, in parental generation, there are yellow round and green wrinkled plants are the combinations. These are the parental combinations. But at the end, in F2 generation, apart from parental generations, we also get two new combination such as green round and yellow wrinkled. That is, yellow character is not tied with the round and green character is not tied with the wrinkle, so that they can separate from each other and forms new combinations such as green round and yellow wrinkle. It concludes that inheritance of one character is independent on another character. So in this way, we have learned the three laws such as law of dominance, law of segregation, and law of independent assortment. I hope you will understand the things. So we will stop here and we'll meet again in next session, which is based on back cross and reciprocal cross. Till then, thank you.